In this lesson, we'll try to make a connection between the psychology of learning and memory that we've examined through experiments and the biology of the brain. What's going on in the brain that allows us to learn things and remember things? We've learned that the surface of the brain is called the cerebral cortex, and humans have a very large brain. So we've got lots of real estate here with which to store information. We've also briefly learned that scientists over the decades have been able to map out the different functions of different regions of the cerebral cortex. So, for example, the back part of the brain here, the surface here, is a visual part of the brain. Here we have the hearing part of the brain and the touch. Here we have movement area of the brain, speech production, language comprehension, etc. Now, how did scientists make this map? Well, there were three basic lines of evidence. Number one, what happens when a certain part of the brain is damaged? If the back part of the brain is damaged, people report blindness. If this part of the brain is damaged, people report that they can't hear, etc. Second line of evidence is just sort of the connections of these brain areas to different sensory systems. For example, the back part of the brain is connected to the eyes. So when our eyes are open and we're collecting light, signals are sent from the eyes along nerves to the back part of the brain. And so this part of the brain becomes active when we're seeing things. So again, second line of evidence that this is visual cortex here is that it's connected up to the eyes and is active when we are seeing. And the third line then is that we can go in with stimulating electrodes and stimulate this area of the brain. And when you stimulate the back part of the brain, people report a visual experience. If you stimulate this part of the brain here, people report hearing something, etc. So those three lines of evidence, brain damage, when is the area of the brain active, what kind of task activates it, and then stimulating that part of the brain, has produced this map that we see here. And if you think about all the functions that are going on in the back part of the brain, it's as if the back part is answering the question, what is happening? So we've got the visual area of the brain, hearing, touch, language comprehension. So the back part of the brain is saying, what's going on? What am I seeing, hearing, feeling? What kind of language is coming my way, etc. The front part of the brain seems to be involved with what should I do. We have regions of the brain that are very active when we're thinking, making judgments, producing speech, making movements with our body, etc. Well, in a school setting then, here's where we want to start to make the connection between psychology and biology. Somehow, depending on what kind of processing tasks we're doing, brains are either poorly remembering the information or successfully storing the information and retrieving it later on. So a shallow processing task just is not allowing the brain to store that information in a way that can be easily retrieved later on, whereas deeper processing tasks are leading to better and stronger and more durable memories that can be more easily retrieved. So there's something different going on in the brain when we're doing different tasks. And so in this lesson, we'll try to see what is it that's happening when we learn some information and then we make a memory and then retrieve it. Well, to get to the bottom of that, we'll have to dig into brains and see what they're made of. They're made of brain cells called neurons, and they are separate living cells, and we can see a bunch of them here. Here's a neuron, there's a neuron, there's one, there's one, there's one, there's one, there's one, there's one. And they make close connections with other neurons. That junction's called a synapse. So when two neurons make contact, we call that a synapse. And that's going to be the way they communicate with each other. So neurons generate electrical signals in response to the activity of other neurons, and then they communicate their own activity to yet other neurons. For example, this one at the top right, it's shown to be sort of illuminated. Now, in reality, brain cells don't generate light, although we're going to see scientists have engineered neurons to do just that. But that we'll take that up in a moment. But when a neuron generates an electrical signal, it then is going to communicate its activity to another neuron by way of the synapse here. So the activity of one neuron can influence the activity of the neuron it's connected to. So when you think about neurons as having many connections, well, they then are going to be sort of getting a pattern of activity from all the other neurons that are connected to it. And that pattern of activity will determine whether that neuron will generate its own electrical signal. So here we see a basic neuron. We have the cell body. We've got these branches off the cell body. Inside the cell body is the nucleus where DNA would be. It's got this long extension called an axon, and then it's got these terminals. And these terminals down here would be the points of connection with other neurons in the brain. We can think of a neuron then as processing information, and there's a flow of information going through the neuron, as it were. For example, the dendrites, these branches here, all these branching structures here are called dendrites, and these are sort of the collection points for information from other neurons. So thousands of synapses would occur at these dendrites. This would be the contacts from many other neurons. So we can think of the dendrites as the receiving zone, the antenna of the neuron. These dendrites are registering activity of all the other neurons that are connected to this single neuron here. The cell body is going to integrate all that activity at synapses there and, in a sense, make a metaphorical decision to generate an output signal or not. So depending on the activity at all those synapses that you don't see here, they're not drawn, the target neuron here, this neuron will then generate its own output electrical signal. That electrical signal runs down the axon here to the terminals, and at the terminals, chemicals will be released here to influence the next brain cell in the chain. Here's another look at a neuron. Like we just saw, we have the cell body here, we have dendrites, but now we can see some synapses from other neurons in the brain. And so the idea here is that there's a combination of processes that allow neurons to function. It's a combination of electricity and chemistry. Electricity in the following way, that a neuron, its signal is carried down its axon as a form of an electrical disturbance. So that's the electricity. The chemistry comes into play when electrical signal reaches a terminal. Chemicals are released, and we can see that back over here. Little chemicals are being released from the terminals of another neuron, and those chemicals can cause small electrical disturbances on the dendrites at that location. And so the idea is this single cell here is sort of receiving information, as it were, from many other neurons simultaneously. That information is in the form of these small electrical signals, and what this neuron then kind of does is it kind of adds up all this activity, and if all that activity reaches a critical threshold, it will generate its own output electrical signal, which of course has the effect of releasing chemicals to influence the next brain cell in the chain. So basically, a neuron is like a little sort of computer chip in a sense. It's got inputs from other neurons, and then it kind of decides what to do with all the activity at its synapses. Should it generate an output signal or not? If kind of we use the analogy of hearing, it's like the neuron is listening to the messages from other neurons, and if the message is important, it will send its own message to the next neuron and all the neurons that it is connected to. Now I want to show you during development, neurons, they physically grow and to reach out and make connections. So in this video, you're going to see neurons that have been placed in a petri dish, and already they have some of the branching structures, some of the dendrites and the axons, but you're going to see over time, this will be time-lapse video, these neurons are growing in the petri dish and reaching out and making contacts with other neurons. This happens during development, but this kind of process also happens when we learn new information. Brain cells are going to be physically growing to make stronger connections and new connections as we learn things. So here's the time-lapse of just sort of neurons in a petri dish with plenty of food and nutrients and oxygen. They're growing and making connections with other neurons. Now we're seeing the growth process here. We're not really seeing the electrical activity of a neuron. And of course we can't see the events at the synapse, it's very small. But this really gets the point across that neurons are living cells. They can grow. And it's this capacity for growth that allows our brains to learn new information, store a memory, and retrieve it later on. Now, I said earlier that neurons do not generate light when they become electrically active. That is until recently. So researchers at some of the top universities in the nation, Stanford and MIT, are developing techniques to actually engineer neurons so that when they make electrical signals, they actually emit light, and that we could potentially visualize. So I'm going to show you a very new technique in microscopy. Here we have a rat here, and let's imagine the rat is kind of immobilized, but it's fully awake and it can sense things, and so maybe it's looking at some stuff in its environment. And what scientists have been able to do is to construct very tiny microscopes to insert down into the brain of the rat. So like us, rats have two hemispheres, the left hemisphere and the right hemisphere. And now if we kind of just take a look at the left hemisphere from the side, we're going to draw it kind of like the human brain that we've been seeing. So here's a left hemisphere. Over here we see this really thin optical fiber. And what scientists have done is put two little lenses on these tiny, really thin optical fibers. And then that's hooked up then. So the optical image would be sent to a camera. So what you can do with this little fiber is insert it into the brain. And you could visualize neurons on the surface of the cortex, or even insert this deeper into the brain to visualize neurons that are at deeper levels. See, that would be very hard with traditional microscope techniques. Now, the other part of this, then, not only of having this tiny, tiny little optical fiber microscope that you can shove anywhere in the brain, is that they have been able to engineer neurons using sort of genetic engineering techniques, engineer neurons to flash light, to release light when they make electrical signals. So here we see some of the neurons in the microscope field of view. What we're about to see, I'm going to show you the actual video here in a moment. What we're about to see are neurons that are flashing. They appear to be flashing. You'll see little flashes of light. That would be a neuron that is currently making an electrical signal. Other neurons then wouldn't be flashing if they're not electrically excited. So over time then, as populations of brain cells, increase their activity or decrease their activity, you're going to just see this kind of population of flashing lights representing the activity of neurons that are currently generating electrical signals. This is what it looks like. So here we see the surface of one part of the cortex of the rat brain, and you can see all these little spots of light. These are neurons that are being electrically excited by whatever neurons were, connected to them. And so we get a sense that the brain is really made up of this huge number, billions of neurons, all electrically excitable, and we're visualizing this pattern of excitation that is happening in the rat's brain at that moment. Now, depending on where we're recording here, right, these could be visual neurons. So these brain cells are responding in a certain pattern of activity to what the rat is currently seeing. And if the rat were seeing something different, a different population of neurons would become active. Let me show you another video like this. And here we're going to see a magnified portion. On the left, again, we see kind of the surface of the cortex here. Notice these big dark things. These are blood vessels. And in fact, you're going to see the movement of blood through the vessels in this portion of the video over here. Let's take a look. So now we see even closer up. The flashing elements here are neurons. And if it flashes, it means the neuron is making an electrical signal. And let's just take a look at the blood vessels real quick. Here we can see blood cells actually traveling through blood vessels. So every neuron in our brain has to be near a blood vessel to get sugar, to get glucose, that's its food, and oxygen. But what's just so fascinating with this technique is to just visualize the activity of brain cells. Again, normally brain cells do not generate light. They generate electrical signals, and then they communicate with other neurons with chemistry at the synapse. But here these neurons have been engineered to flash light, so we can see those neurons at any one time that happen to be active, processing whatever information is going on in that region of the brain. So again, if this were visual cortex, then whatever the rat is looking at, it's these populations of neurons that are responding. Now, we're not seeing all kinds of other neurons that are just not currently active, given what the rat is seeing at the moment. If the rat were seeing some other different kind of object or some different scene, some different environment, a different population of neurons would be activated. So we're sort of peering in and seeing the brain in a very new way. We're seeing what brains are doing when we are seeing things and when we're thinking. It's electrically excitable cells are generating electrical signals, but those signals have been converted into flashes of light so we can better appreciate what's going on in a brain. So with that introduction now, we're ready to, in part 2, we'll take a look at what's really going on when we acquire information, we learn this information, we make a memory, and then we retrieve it. So we're going to break that process down into 3 stages. Encoding, sort of when we're processing the material we're learning. Storing, we're going to store some kind of memory of the information, and then we're going to retrieve that information at a later time. So we'll pick that up in part 2.